GRAPE is an open-source genomic relatedness detection pipeline for genetic testing companies and research institutions. Ancestry prediction and DNA relatives matching services have been the key drivers for market growth and development since the emergence of the direct-to-consumer genetic testing market. These services accounted for more than 50% of 30 million people who had taken genetic tests to date. Such demand can be considered a social phenomenon that embraces people's eager desire to explore their origins and connections worldwide. It is, therefore, no surprise that these services are best-selling among all major personal genomics companies. The key component of any DNA matching service is a data processing pipeline that can identify human relationships from genotypic data. However, assembling a pipeline that reliably operates on real-world genotypic data requires a significant amount of research and development resources. Companies can spend more than a year building and integrating DNA matching pipelines into their services. The pipeline must combine necessary pre-processing steps and a comprehensive data quality control to work with real-world datasets. It should be accurate for both close and distant degrees to provide meaningful results, and should be flexible enough to support various production applications. GRAPE is a genomic relatedness detection pipeline under the GPL v3 license. GRAPE is a reliable and accurate end-to-end software that is free for both commercial and academic purposes. It was developed in collaboration with a global personal genomics company, Atlas Biomed Group. GRAPE predicts a relationship degree for a given pair of individuals using genetic data. Every person inherits half of their parental DNA in chunks. Then, some of these chunks are passed on to the next generation, possibly broken, during the recombination process. If two people have identical chunks of DNA inherited from the same common ancestor, they are relatives. These chunks are called identical by descent segments. Close relatives have more identical chunks in common with higher total and average lengths. This information lies behind predicting a relationship degree between two individuals. The pipeline adapts software development best practices such as using a snake-make workflow management system, Docker containerization, and GA4GH standards and tools. GRAPE requires a single multi-sample VCF file as input and is easy to use. It has a separate workflow for downloading required reference datasets and checking their consistency. As real-world datasets are often heterogeneous and inconsistent, the pipeline has various pre-processing and quality control options. These steps are essential for ensuring accurate outcomes and low false positive rates. The pipeline pre-processing includes optional phasing and imputation. It supports input data in both HG38 and HG19 assembly and allows conversion between them. During the quality control stage, GRAPE removes all indels and rare SNPs, checks for strand and SNP ID mismatches, and removes all suspicious samples, namely with high missingness and homozygosity. To perform relatedness estimation for a given pair of samples, the pipeline should previously detect IBD segments. Having tested the majority of available tools, we have chosen IBIS as the most accurate and efficient IBD segments detection software. IBIS can work with unphased data. It allows controlling false positive rate and sensitivity by adjusting IBD inference parameters. Other available tools such as King and Plink work with population allele frequencies. King is a widely known tool for determining close degrees of kinship and is optionally available in the GRAPE pipeline. URSA tool is a gold standard for estimating the most probable degrees of kinship based on IBD statistics. URSA uses the method of maximum likelihood to guess the most probable relationship degree from the length and the total number of IBD segments detected between two individuals. You can see the equations for the length and total number of shared IBD segments distribution, which are used by URSA on the left, and an example of the total IBD length distribution among the different degrees on the right. GRAPE's detection power. GRAPE supports the adjustment of tools for better control of precision and recall levels. To perform the precision recall analysis of the pipeline, we used PED-SIM simulation software to generate a few pedigrees from unrelated individuals using genotypic data from the 1000 Genome Project. The method uses sex-specific genetic maps to simulate pedigree structures. The left panels demonstrate the metrics for the pipeline with the strict control over the false positive rate. The right panel shows a confusion matrix. Pipeline operators can relax parameters of the GRAPE pipeline to receive more matches and increase the recall level among higher degrees. However, in this case, the number of false positive matches increases, which can be seen on the confusion matrix in the right panel. Thanks to the comprehensive pre-processing module, GRAPE is very reliable and can produce almost zero false positives when needed. GRAPE is ready to operate on highly diverse real-world datasets because of its performance and flexibility with the settings and parameters. This is an example of the main GRAPE report. GRAPE produces IRSA confidence intervals for each degree and the basic IBD statistics. Output is produced for every pipeline step, including all IBD segment data. Logging occurs for each launch with an entry in the message log. It also shows king degrees and kinship when king is used. The pipeline is a free and open source project under the GPL v3 license. The pipeline uses software distributed under open source licenses. The only exception is King, as it requires written author permission for non-academic purposes. However, King is optional and can be fully interchanged with IBIS and IRSA tools. The performance analysis confirms that the pipeline can handle 10K samples with the primary IBIS-only option in half an hour. GRAPE can utilize multiple cores and is currently suitable to handle datasets of around 100K samples. GRAPE is a user-friendly, fast, and accurate software product. We welcome you in our GitHub repository and support chat. Thanks for your attention.